Transcription. Transcription involves the synthesis of an RNA chain. Transcription takes place by the usual process of complementary base pairing, catalyzed and scrutinized by the enzyme RNA polymerase. The RNA chain transcript is synthesized from the 5' end towards the 3' end. The transcript is identical in sequence with one strand of DNA, which is called coding strand, and the complementary to the other strand called template strand or antisense strand. So, transcription unit. Each transcribed segment of DNA is called transcription unit. In eukaryotes, a transcription unit typically carries the information of just one gene and therefore coded for either a single RNA molecule or a single protein. In bacteria, a set of adjacent genes is often transcribed as a unit. The resulting mRNA molecule therefore carries the information for several distinct proteins. The immediate product of transcription is called the primary transcript. Transcription units may be simple or complex. The primary transcript produced from the simple transcription unit is processed to yield a single type of mRNA, coding a single protein. In the case of complex transcription units, which are quite common in multicellular organisms, the primary RNA transcript can be processed in more than one way, leading to formation of more than one type of mRNAs. Transcription starts from the first base pair, that is called the start point. From the start point, RNA polymerase moves along the template, synthesizing RNA until it reaches a terminatal sequence. So, sequences prior to the start point are described as upstream of it. Those after the start point within the transcribed sequence are downstream of it. Next is about prokaryotic transcription, RNA polymerase. RNA synthesis is catalyzed by the enzyme RNA polymerase. A single type of RNA polymerase appears to be responsible for almost all synthesis of mRNA and all rRNA and tRNA in a eukaryotic eubacterium. No, we are talking about prokaryotic eubacterium. There is no eukaryotic eubacterium, just prokaryotic eubacterium. I'm sorry for the mistake. And eubacterial RNA pol is a multi-subunit enzyme made up of 4 different polypeptides: alpha, beta, beta prime, and sigma. The holoenzyme can be separated into 2 components: the core enzyme and the sigma factor. The complete enzyme or holoenzyme in E. coli has a molecular weight of about 465 kilodalton. The alpha subunit is required for assembly of the core enzyme and plays a role in promoter recognition. The alpha subunit also plays a role in the interaction of RNA polymerase with some regulatory factors. The beta and beta dash subunits together make up the catalytic center. The beta subunit involves the chain elongation. The sigma subunit is concerned specifically with the promoter recognition. The catalytic activity of E. coli RNA polymerase is provided with a heterotetramer composed of beta and beta dash or beta prime subunits and 2 copies of small alpha subunit. The process of transcription. The transcription reaction can be divided into 3 stages, like initiation, elongation, and termination. Transcription begins when an RNA polymerase molecule binds to promoter sequence on the DNA double helix. The polymerase then moves along the template strand, extending its growing RNA chain in the 5' to 3' direction by the stepwise addition of ribonucleoside triphosphates until it reaches a termination signal, at which point the newly synthesized RNA chain and polymerase are released from the DNA. Then initiation, it describes synthesis of the first nucleotide bonds in RNA. The sequence of DNA needed for RNA polymerase bind to the template and accomplish the initiation reaction defines the promoter. And promoter, a gene promoter, can be defined as a cis-active DNA sequence necessary for accurately and efficiently initiating transcription of the gene. The DNA sequence of promoter region is recognized by the requisite, that is RNA polymerase. The best characterized bacterial promoter are those of Bacterium E. coli that are recognized by sigma nd. The superscript indicate molecular weight. We represent like this. The superscript is something which is usually represented at the top. It contains 2 6 base pairs consensus sequence. 10 sequence is also known as Pribnov box. The 10 sequence has the consensus TATAAT and the 35 sequence has consensus TTGACA. The 10 and 35 regions are so designated to reflect their approximate distance in nucleotides from start site. The distance separating the 35 and 10 sites is between 16 and 18 base pair and 10 hexamer, lying 7 base pair upstream of the start point. Upstream, downstream, 35 region, consensus sequence, then consensus sequence is also there. So bacterial promoters such as that from E. coli shown here share two regions of highly conserved nucleotide sequence. These regions are located 35 and 10 base pair upstream from the start site of transcription, which is indicated as +1. By convention, all nucleotide upstream of the transcript's initiation site at +1 are numbered in a negative sense. These elements function only in a double-stranded DNA. And consensus, the sigma subunit enables RNA polymerase to recognize promoter sites. Most E. coli promoters interact with the main form of RNA polymerase, which contain sigma 70. Sigma 70 in the sense its molecular weight. Okay. So transcription of certain groups of genes, however, is carried out by E. coli RNA polymerase containing one of several alternative sigma factors. These alternative sigma factors like sigma 28, sigma 32, sigma 38, and sigma 54 recognize different consensus promoter sequences than sigma 70. Nitrogen assimilation, major sigma factor during stationary phase. Also for genes involved in oxidative and osmotic response, heat shock response for genes involved in flagella synthesis. The holoenzyme promoter reaction starts by forming a closed binary complex. Closed means that the DNA remains duplex, and the RNA polymerase holoenzyme initially binds to DNA. It covers some 75 to 80 base pair, extending from -55 to +20. The closed complex is converted into an open complex by melting a short region of DNA within the sequence bound by the enzyme. The transcription bubble is created by the local unwinding that begins at the site bound by RNA polymerase. The length of the transcription bubble created by a local unwinding is about 12 to 14 base pair, and the length of RNA DNA hybrid within this is 8 to 9 base pair. The next step is to incorporate the first two nucleotides. Then a phosphodized bond forms between them. This generates a ternary complex that contains RNA as well as DNA and enzyme. Initiation describes synthesis of first nucleotide bonds in RNA. Only the holoenzyme can initiate transcription. Core enzyme has the ability to synthesize RNA on a DNA template but cannot initiate transcription at the proper sites. So RNA synthesis is frequently about after usually 2 to 3 but up to 10 to 12 nucleotides have been joined. This phenomenon is known as abortive initiation. Once a polymerase manages to make the RNA longer than 10 base pair, a stable ternary complex is formed. This complex contains the enzyme, the DNA template, and a growing RNA chain. Then control of transcription initiation. In bacteria such as E. coli, there are two distinct ways in which transcription initiation is controlled. Constitutive control, which depends on the structure of the promoter. Then regulatory control, which depends on the influence of regulatory proteins. Promoter structure determines the basal level of transcription initiation. The consensus sequence for the E. coli promoter is quite variable. Different promoters vary in their efficiency. Efficiency is defined as the number of productive initiations that are promoted per second. The productive initiation being one that results in the RNA polymerase clearing the promoter and beginning synthesis of a full-length transcript. It may be strong or weak promoter. Strong promoters support high rate of transcription initiation. So RNA polymerase, binding of RNA polymerase. Then closed complex, open complex, transcription initiation, promoter clearance and elongation for transcription initiation and elongation by E. coli RNA polymerase. The enzyme first forms a closed complex in which the two DNA strands remain fully base paired, and the next step the enzyme catalyzes the opening of the DNA helix to form an open complex in which the template DNA strand is exposed for the initiation of an RNA chain. The polymerase containing the bound sigma subunit. The conversion to an actively elongating polymerase requires the release of initiation factors, the sigma subunit. Later is elongation. The elongation involves the movement of transcription bubble by the disruption of DNA structure. The enzyme moves along the DNA and extends a growing RNA chain. As the enzyme moves, it unwinds the DNA helix to expose the new segment of template in single-stranded condition. Nucleotides are covalently added to the 3' end of the growing RNA chain, forming an RNA-DNA hybrid. During each nucleotide addition, the beta and gamma phosphates are removed from the incoming nucleotide and the hydroxyl group is removed from the 3' carbon of the nucleotide present at the end of the chain. The overall reaction rate is 40 nucleotides second at 37 degrees Celsius. During elongation, when RNA polymerase transcribe DNA, unwinding and rewinding occur. As RNA polymerase move forward along the double helix, it generates positive supercoils ahead and leaves negative supercoils behind. Then enzyme gyrases and topoisomerase 1 participate during this process. Then gyrase introduces negative supercoils, whereas topoisomerase 1 removes negative supercoils. Then cordycepin, an adenosine analog that lacks a 3' OH group, inhibits elongation. Its addition to the 3' end of RNA prevented RNA chain's further elongation. Here we could see cordicipin 3-deoxyadenosine 3' deoxyadenosine. And last one is the termination process. It involves recognition of the point at which no further bases should be added to the chain. The sequence of DNA required for this reaction is called terminator. At this point, the enzyme stops adding nucleotides to the growing RNA chain, releases the completed product, and dissociates from the DNA template. Exactly how termination occurs is not known. Bacteria appear to use two distinct strategies for transcription termination: intrinsic termination and row-dependent termination. So intrinsic termination in the sense have been, so direction of transcription is to the right side, then inverted repeats are there, high G plus C content, then template stand, last base transcribed, and loop formation is also there. The E. coli RNA polymerase stop when it synthesizes a turn of U residues from the complementary run of A residues on the template strand. A self-complementary RNA nucleotide sequence rapidly forms hairpin helix, which is crucial for stopping transcription. The second type of termination is row dependent. This termination requires the activity of protein called row. Row is an ATP dependent ATP stimulated helicase that disrupts the nascent RNA DNA complex. It is a hexamer of identical subunits. It binds to RNA and translocates along RNA unit. It reaches the RNA DNA hybrid in RNA polymerase, where it releases the RNA from the DNA. What is the minimum length of time required for the synthesis of E. coli polymerase of an mRNA encoding a 100 kb protein? A 100 kb protein contains about 9-10 residues which are encoded by 2730 nucleotides. At a maximum transcription rate of 50 nucleotides per second, the protein would be synthesized in 54.6 seconds. Why is RNA synthesis is not as carefully monitored for errors as in DNA synthesis? An error will only affect one molecule of mRNA of many synthesized from a gene. In addition, the errors do not become permanent part of genomic information. Di and trinucleotides are occasionally released from DNA polymerase at a restart of transcription, a process called abortive initiation. This process requires the restart of transcription, suggests a possible explanation for abortive initiation. The base pairing energy of the di and trinucleotide of DNA RNA hybrids formed at the very beginning of transcription is not sufficient to prevent standard separation and loss of product. Next is about eukaryotic transcription. Nuclear RNA polymerase. A single RNA polymerase is responsible for transcription of all different types of RNAs in prokaryotes and archaebacteria. However, eukaryotes have 3 RNA polymerases. In addition, an additional RNA polymerase is found in mitochondria as well as in chloroplast, which carry a small DNA molecule of their own. RNA polymerase 1 is responsible for synthesizing most of RNA. Polymerase 2 synthesizes mRNA and most of the snRNA, and polymerase 3 synthesizes a variety of small stable RNA including tRNA, 5sRNA, and U6 snRNA. Most RNA polymerase is multi-subunit protein, 8 to 12 subunits. Eukaryotic polymerase 3 consists of 12 subunits. The two largest subunits are homologous to the bacterial beta and beta dash subunits. In addition to the increased number of subunits, eukaryotic polymerase 2 differs from its prokaryotic counterpart in that it has a series of heptad repeats, which with consensus sequence given at the carboxyl terminal of the target polymerase 2 subunit. This carboxyl terminal repeat domain, or CTD, is both a substrate for several kinases, including the kinase component of TF2H and the binding site for a wide array of proteins. An RNA polymerase 1 product. Polymerase 1 synthesizes only a single RNA species, the large ribosomal RNA precursor molecule. The large rRNA precursor molecule is processed into mature 5.8S, 18S, and 28S rRNA. RNA polymerase 2 product, mRNA, most of snRNAs. Translational adapter, ribosomal component, mRNA splicing, rRNA-P component, rRNA splicing, signal recognition particle component, some unknown particles are also there. Then, RNA polymerase in eukaryotes display different sensitivities to toxin called alpha-amanitin, a cyclic octapeptide that is produced by the poisonous Amanita mushrooms. Alpha-amanitin works by interfering with the translocation process during RNA elongation. Pol 2 is the most sensitive to alpha amanitin, and Pol 1 is completely resistant. Then eukaryotic promoter. In eukaryotes, the term promoter is used to describe all the sequences that are important in the initiation of transcription of a gene. For some genes, the sequences not only includes the core promoter, sometimes called the basal promoter, which is the site at which initiation complex is assembled, but that also one or more upstream promoter elements, which, as the name implies, lie upstream of the core promoter. So RNA pol 1 promoter, RNA polymerase 1 promotes consists of core promoters spanning the transcription start point between nucleotides 45 and 20, and an upstream control element or UCE about 100 BP further upstream. So RNA polymerase 1 promoter, UCE, core promoter, and ribosomal DNA. Transcription units for RNA polymerase 1 have a core promoter separated by about 70 base pairs from the upstream control element. RNA polymerase 1 binds to the core promoter. In RNA polymerase 3 promoter, the most striking and unusual feature of promoters used by polymerase 3 is that the majority include important sequence elements downstream of the transcription start site within the transcribed region. So type 1 promoter found in the 5S rRNA gene require 3 internal elements for efficient transcription and are A block located between the plus 50 and plus 70 and C clock from plus 80 to plus 90. Type 2 promoter found in the tRNA genes consist of 2 highly conserved sequence blocks A negative and B negative located within the transcription region. The first eukaryotic promoter to be characterized belongs to tRNA. Type 3 promoter found in the U6 snRNA genes consist of TATA box or TATA box located between minus 30 and minus 25, a proximal sequence element between minus 66 and 46, and the distal sequence element or DSE between minus 244 and 214. Type 1 internal, type 2 internal, upstream type, and UC small nuclear region transfer 5S ribosomal, etc. So the promoter of RNA polymerase 3 may consist of bipartite sequence downstream of the start point with box A separated from either box C or box B as well as upstream sequences. So 5S rRNA genes have type 1 promoter, tRNA genes have type 2 promoter, and U6 snRNA genes have upstream type promoter. In RNA polymerase 2 promoter, RNA polymerase 2 promoters are variable and can stretch for several kilobases upstream of the transcription start site. The core promoter consists of 2 segments, the TATA box and the initiator sequence. Initiation sequences are rich in pyrimidines. Some genes transcribed by RNA polymerase 2 have only one of these 2 components of the core promoter, and some surprisingly have neither. The later are called null genes. Apart from the 2 core promoter elements, the TATA box and the INR sequence that serve as specific binding sites for general transcription factors, either cis-acting sequence serve as binding sites for a wide variety of regulatory factors that control the expression of individual genes. The cis-acting regulatory sequences are frequently, though not always, located upstream of the TATA box. For example, 2 regulatory sequences that are found in many eukaryotic areas are CCAT box and GC box and CCAAT box and GC box. Specific proteins that bind to the sequences and stimulate transcription have since been identified. Then RNA pol 2 promoter, enhancer, core promoter, TATA, protein encoding regions. RNA pol 2 core promoter consists of TATA box and initiator element. And transcription initiation with RNA polymerase 2 we are going to learn. So transcription factors, unlike bacterial RNA polymerase, purified eukaryotic RNA polymerase 2 could not initiate transcription in vitro. So transcription of eukaryotic genes require many protein factors called transcription factors apart from RNA polymerase to initiate as well as to regulate transcription. So transcription factor is a transregulator. Transcription of eukaryotic genes require many protein factors called transcription factors apart from RNA polymerase to initiate as well as to regulate transcription. Transcription factor is a transregulatory factor, usually a soluble protein produced at a gene distant from the chromosome region that it affects. It attaches to DNA at a gene regulatory site and by so doing influences the rate of transcription of a specific gene. So transcription factors come in 2 general ways: basal general transcription factors or TF2D, TF2A portions, then which attach to gene promoters and cell-specific transcription regulatory factors which bind to regulatory sequences like enhancer, silencer. Transcription regulatory proteins factors may be repressor or transcription activators. The transcriptional activators like SP1 bind to regulatory DNA sequences and stimulate transcription. And transcription initiation by RNA polymerase 2. Gene is transcribed by RNA polymerase 2. First binds with the general transcription factor GTF, general transcription factor, involved in transcription from all polymerase 2 promoters and constitute part of the basic transcription machinery. TF2D, which is a complex made up of data binding protein and at least 12 TBP associated factor or TAFs. TBP is a sequence specific protein that binds to DNA via its unusual TBP domain, which makes contact with the minor groove in the region of the TATA box. During transcription, TAFs assist in attachment of TF2D to TATA box. After TF2D has attached to the core promoter, the pre-initiation complex is formed by attachment of the remaining GTFs. This GTFs bind to the complex in the order TF2A, TF2B, TF2F, and RNA polymerase 2, TF2E, and TF2H. The final step in assembly of initiation complex is the addition of phosphate groups to the C-terminal domain or CTG of the largest subunit of RNA polymerase 2. Once phosphorylated, the polymerase is able to leave the pre-initiation complex and begin synthesizing RNA. So, phosphorylation might be carried out by TF2H. So, functions of human general transcription factor, TF2D or TBP component, recognition of data box and possibly INR sequence forms a platform for TF2B binding. Then TF2D or TAF, recognition of the core promoter, regulation of TBF binding, and stabilizes TB2A, stabilizes TBP and TAF binding. Then TF2B, intermediate in recruitment of RNA polymerase 2, influences selection of the start point for transcription. Then recruitment of RNA polymerase 2, intermediate in recruitment of TF2H, modulates the various activities of TF2H. Helicase activity responsible for the transcription, for the transition from the closed to open promoter complex, possibly influence promoter clearance by phosphorylation of C-terminal domain of the largest subunit of RNA polymerase 2. In this picture, TF2D binds with the data box, then TF2A, TF2B, TF2F and RNA polymerase 2, TF2E and TF2H. Initiation of transcription. Assembly of RNA polymerase 2 pre-initiation complex. The indicated general transcription factors and the purified RNA polymerase 2 bind sequentially to data box DNA to form a pre-initiation complex. In the first step, TF2D binds specifically to a data sequence. Next, TF2B enters the complex, followed by RNA polymerase 2 escorted by TF2F. So TF2E and TF2H then assemble into the complex. In the presence of ATP, TF2H phosphorylates pole 2 initiate transcription. The site of phosphorylation is CTD, which extends from the largest subunit of pole 2. The CTD contains a structure of 7 amino acids and is repeated multiple times. And transcription initiation in a eukaryotic cell is more complex process. Transcription initiation requires the presence of transcription activator proteins. These proteins bind to specific sequence in DNA. A typical eukaryotic gene has many activator proteins which together determine its rate and pattern of transcription. Sometimes acting from a distance of several thousand nucleotide pairs, these genes regulate proteins, help RNA polymerase, the general transcription factors, chromatin remodeling complex, and mediator all to assemble at the promoter. The mediator allows the activator proteins to communicate probably with the polymerase 2 and the general transcription factors. So, gene activator protein binds to chromatin modeling complex, chromatin remodeling, histone modification enzymes, covalent histone modification, other activator proteins, additional activation proteins bound to gene regulatory region, gene transcription factors, RNA polymerase, assembly of pre-initiation complex at the promoter, then rearrangement of proteins in the pre-initiation complex, then transcription initiation. An order of events leading to transcription initiation at a specific promoter in Saccharomyces cerevisiae. The order of steps in the pathway to transcription initiation appear to be different for different promoters. Then enhancers. Many genes in mammalian cells are controlled by regulatory sequences located further away, sometimes more than 10 kilobases from the transcription start site. These sequences may be enhancers or silencers. Enhancers are positive transcriptional control elements. They serve to increase the basal level of transcription, which is initiated to the core promoter element. A typical enhancer is 100 to 300 base pair long and essentially consists of an array of binding sites. The DNA binding transcription factors. In 1981, the first enhancer characterized was from the genome of simian virus 40. Later on, the first cellular enhancer was discovered in the immunoglobulin heavy chain G. Although enhancers were first identified in eukaryotic cells, they were subsequently been found in bacteria. Characterization of enhancers from SV40 and other viruses lead to the following definition of transcription enhancers. It is a DNA sequence, typically about 200 base pairs in size, that strongly activates transcription of a linked transcription unit from its current initiation site. It activates transcription independently of the orientation, and it's able to exert its function over large distances for more than 1000 base pair from a position either upstream or downstream to the initiation site. Enhancer stimulates transcription in a cell type specific manner. Transcription factors bound to distant enhancers can thus work by the same mechanism as those bound adjacent to promoters. And there is no fundamental difference between the actions of enhancer and those of cis-acting regulatory sequences adjacent to transcription start site. If an enhancer activates a specific gene in position and orientation independent manner, then what stops from activating other genes. It is a specific DNA element called insulator control the actions of activator. When an insulator is placed between an enhancer and a promoter, it prevents the enhancer from activating the promoter. This may explain how the action of an enhancer is limited to a particular promoter. So enhancer promoter transcription on, enhancer insulator promoter transcription off. An enhancer activates a promoter in its vicinity but may be blocked from doing so by an insulator located between them. Silencers, control agents. Once again, enhancer promoter. Enhancer insulator promoter transcription of off. Because insulator is there, so transcription is off. Silencers are control agents of DNA that, like enhancers, may be located thousands of base pairs away from the gene they control. Silencers serve to reduce transcription levels. Two classes of silencers have been distinguished: classical silencer and position independent elements, whereas negative regulatory elements are position dependent elements. And role of activator and co-activator. Activator is a DNA binding protein that stimulates the transcription initiation. Some activators recognize upstream promoter elements and influence transcription initiation only at the promoter to which these elements are attached. The other activators target sites within enhancers and influence transcription of several genes at once. In general, these factors have been found to consist of two domains. One region of the protein specifically binds DNA binding domains, and others called activation domains activates transcription by interacting with other components of the transcriptional machinery. The basic function of the DNA binding domain is to anchor the transcription factor at the proper site on DNA. The activation domain then independently stimulates transcription by interacting with other proteins. Diverse nature of activation domains have been reported. Some most common types of activation domains are acidic domains, glutamine-rich domains, and protein-rich domains. One common example of eukaryotic activator is GAL4. It activates transcription of GAL1 gene in the yeast Saccharomyces cerevisiae. GAL4 is normally responsible for activating the transcription of yeast genes that code for the enzyme that convert galactose to glucose. GAL4 binds as a dimer to the 17 base pair site on DNA located 275 base pair upstream to GAL1. The GAL4 protein found to DNA in the rough vicinity of the promoter facilitates addition of TF2B to the nascent complex of general transcription factors. When found, it activates transcription of GAL1 gene manifold. Then DNA binding motifs. DNA binding proteins such as transcription factors recognize and bind a short nucleotide sequence usually as a result of extensive complementarity between the surface of the protein and surface features of the double helix in the region of binding. A DNA binding motif permits specific binding of the DNA binding protein to its target sequence. Several conserved structural motifs have been identified, which are common to many different DNA binding proteins with quite different specificities, including the leucine zipper helix, loop helix, and helix turn helix, along with zinc finger motifs. Then, helix turn helix motif. A helix turn helix motif is present in many eukaryotic and eukaryotic DNA binding proteins. The helix turn helix motif was the first DNA binding structure to be identified. The motif is made up of two alpha helixes separated by a beta turn made up of 4 amino acids, the second of which is usually glycine. In turn, this turn in conjunction with the first alpha helix positions the second alpha helix on the surface of the protein in an orientation that enables it to fit inside the major groove of an DNA molecule. The second alpha helix is known as recognition helix. The HTH or helix loop helix, helix turn helix, we are learning about helix turn helix inside the major groove of DNA molecule. Structure is usually 20 or so amino acids in length, and so it is just a small part of protein as a whole. Many prokaryotic and eukaryotic DNA binding proteins utilize the helix turn helix motif. Recognition helix, COOH and NH2 is there. So helix turn helix motif, the C-terminal alpha helix is called the recognition helix because it participates in sequence-specific recognition of DNA. This helix fits into the major groove of DNA pairs. The homeodomain is an extended helix turn helix motif presented by each of these proteins. It is made up of highly conserved domain of 60 amino acids encoded by a 180 base pair sequence called a homeobox, which form 4 alpha helixes, numbers 1, 2 and 3 separated by a beta turn, which number acting as a recognition helix and number 1 making contacts with the mean of 2. And helix loop helix. The helix loop helix domain is involved in protein dimerization. The helix loop helix motif is composed of two regions of alpha helix separated by a region of variable length which forms a loop between the two alpha helixes. This motif is quite similar to the helix turn helix motif found in different prokaryotic transcriptional factors such as CRP protein involved in the regulation of lac operon. The HLH motif mediates both DNA binding and protein dimer formation. It permits occasional heterodimer formation. So, active HLH homodimer. The HLH motif is responsible for both dimerization and DNA binding. Then, leucine zipper motif. The leucine zipper is 30 to 40 amino acids long with a leucine at every 7th residue to form a heptad repeat. The zipper region forms an amphipathic alpha helix with the leucines and other hydrophobic residues arranged along one surface. The leucine zipper domain is necessary for protein dimerization. It is a motif generated by the repeating distribution of leucine residues spaced 7 amino acids apart within an alpha helical regions of the protein. So these leucine residues end up with the R groups protruding from the alpha helix domain in which the leucine residues reside. The protruding R groups are thought to interdigitate with the leucine R groups of other leucine zipper domain, thus stabilizing the homo or hetero dimerization. The leucine zipper domain is present in many DNA binding proteins such as CMYC and CEBP. So two large alpha helixes, one from each monomer, form from the dimerization and DNA binding domain at different sections along their length. Then zinc finger means zinc coordinated DNA binding motifs are called zinc fingers. There are several types of DNA fingers. The most widely occurring zinc finger motif in eukaryotes is the C2H2 zinc finger, which was first discovered in transcription factor called TF3A that regulate the genes for 5S rRNA. Proteins of the steroid thyroid hormone family of transcription factors also contain zinc fingers. This class of zinc fingers occur in clusters Each finger is 30 amino acids long and is characterized by pairs of cysteines and histidines that always occur in the same relative positions. It forms a compact globular domain in which a zinc ion is coordinated tetrahedrally by the cysteine and histidine residues. The zinc is essential for correct folding and for DNA binding. NMR and X-ray studies have shown that the C2H2 finger is a compact globular domain composed of a 12 residue alpha helix packed against an irregular beta sheet with a zinc in between. The alpha helix of each finger lies in the major groove and contacts 3 base pairs. The base contacts are almost entirely with one DNA strand. The second class of DNA binding domain that makes structural use of zinc is found in the nuclear receptors, a superfamily of ligand-activated transcription factors that mediate the response to intracellular stimuli, such as steroid hormones, thyroid hormones, retinoic acid, and vitamin D. The DNA binding domains of these factors are 70 to 80 amino acids long and contain 2 zinc ions which coordinated tetrahedrally with 4 cysteine residues. They are referred to as C2C2 zinc fingers. These domains contain 2 similar loop helix motifs that are folded together to form a single structural unit. Each motif has a zinc ion ligand by 2 cysteines at the start of the loop and 2 cysteines at the end terminus of the alpha helix. Helix turn helix, zinc finger, and leucine zipper. Then antibiotic inhibitors of transcription. So rifampicin and actinomycin are 2 antibiotics that inhibit transcription. So rifampicin is a semisynaptic derivative of rifamycin. Then rifamycin binds in a pocket of the beta subunit and blocks the path of the elongation RNA. By preventing the RNA chain from extending beyond 2,3 ribonucleotides, this blocks transcription. Actinomycin D binds tightly and specifically to double helix DNA and thereby prevents it from being an effective template for RNA synthesis. It does not bind to single-stranded DNA or RNA, double-stranded RNA or RNA-DNA hybrids. The phenoxazone ring system of actinomycin D intercalates between adjacent G to C base pairs because it is strongly heterophobic and its dimensions permit it to slip into the space between the pairs with minimum deformation of the double helix. The 2 cyclic peptide structures bind to the minor groove of the double helix. The deformation of the helix caused by the intercalated actinomycin D prevents the movement of RNA polymerase along the DNA template and thus inhibits RNA synthesis. It is extensively used as a transcription inhibitor for the formation of new RNA in both prokaryotic and eukaryotic cells.